Okay, good morning, good afternoon, good evening, everyone. Welcome to the ZGU Earth Data webinar. Today it's our great pleasure to have Dr. Ben Uveges speaking with us from Cornell. So Ben got his undergraduate bachelor's degree in chemistry, started from McGill, and then got his PhD from Syracuse. After that, he has been conducting his postdoctoral research in MIT. That's basically the work of the talk they're coming from. And now he's currently based in Cornell. And Ben is interested in stable isotope geochemistry and organic geochemistry. And he has broad interest in biogeochemical cycling of nutrients, depositional characteristics of anoxic marine basins, and the use of molecular biomarkers for both the paleoenvironmental and ecological studies. So today, Ben is going to share with us about one part of his research of this paleo rocks based on 4 drilling cores from the carbon isotopes and nitrogen isotopes, and let us review the nitrogen cycle before the great oxidation event. And with that, Ben, there we go.
Well, thank you very much for that wonderful introduction. And I want to say thank you all for having me. It's very nice to get the invite. So as was alluded to here, I'm going to talk to you today about the onset of the aerobic nitrogen cycle and how that came about and the timing of it relative to the oxygenation of the atmosphere. And as I kind of go through this talk, I'm actually going to take this title and go backwards. So I'm going to start with oxygen, because really when you're talking about the nitrogen cycle, it's really intimately and intrinsically linked to oxygen, right? It's hard to, you can't talk about one without the other. And so we're going to start with trying to understand the timing of permanent atmospheric oxygenation, and then we'll move into the impact on the nitrogen cycle and the timing of that and how this kind of all fits together. So why are we going to talk about oxygen? Well, telling the story of oxygen is kind of like telling our collective eukaryotic history, right? The rise of oxygen profoundly impacted the Earth's surface. We go from this largely reducing world back, prior to say 2.5 billion years ago, to this transition across what's known as the Great Oxidation Event, or GOE, to our modern atmosphere, where about 1/5 of it is oxygen, right? So this really, really stark and dramatic transition across Earth history. There are a number of proxies that point towards the early production of oxygen. So the evolution or advent of oxygenic photosynthesis. Now, I'm not going to wade into the debate on what the timing of that is, because I know that's a thorny subject for many people, but safe to say it's somewhere between, say, 3.2 and 2.5 billion years ago. There are a number of competing ideas about the timing of the accumulation of the oxygen, or the product of that photosynthesis, in the atmosphere, right? So you can see these two different kind of broad interpretations of our atmospheric oxygen concentration, the orange and the blue here. And understanding and constraining the nature of that transition, the timing of that transition, is important for understanding of the evolution of complex life and the Earth system, right? And so when I say the evolution of complex life, I'm mostly talking about eukaryotes, so complex multicellular life. And eukaryotes and complex life as we know it needs oxygen, right? So we need to have oxygen in some appreciable levels, like what we have today, in order for this, incredible ecological diversity within eukaryotes that we know to exist. And this extends beyond just the requirement for oxygen for the type of respiration that eukaryotes undergo. It also is a fundamental control on their biochemistry or structure, right? So if we look at the production of sterols, or at their geological derivatives, steranes, the production of sterols by eukaryotes, which these are their kind of typical membrane lipids, requires about 11 moles of oxygen per mole of sterol produced, right? So even just the building blocks of our cellular materials require oxygen. So we're really steeped in this requirement. So we're going to talk about what we know about the timing and the nature of the Great Oxidation Event. And broadly speaking, we can pin down when this transition occurred based on the rock record, right? So if we look at the presence of oxygen-sensitive detrital grains, known such as uranite or pyrite, they disappear about 2.5 billion years ago. We don't really see those as detrital grains anymore. We also see the rise and fall of banded iron formations, these formations that are kind of dependent on this periodic redox cycling of iron. And then we also see the advent of manganese deposits, oxidized iron in paleosols and red beds, and sulfate deposits. So this transition from a reduced sulfur world known as sulfide to sulfate. And all of these things kind of coalesce around this gray box here, which is somewhere between, say, 2.3 and 2.5 billion years ago. Now, our most direct proxy for what's actually happening in the atmosphere, or pinning down these concentrations that you see on the y-axis up here, is what's known as the mass-independent fractionation of sulfur isotopes. So these data points here, or which is represented as CAP33S in per mil notation. And just to kind of give you a brief primer on what CAP33S actually is, this was representing the mass-independent fractionation of multiple sulfur isotopes. It was first discovered about 25 years ago now by James Farquhar and colleagues. And as you can see here across Earth history on the x-axis here, from about 4 billion to modern, we see this presence of appreciable CAP33S values between, say, -1.5 to 2, and then a disappearance of it across this kind of 2.5 to 2.3 GA boundary. Now, that CAP33S is defined as the deviation of multiple sulfur isotopes from expected, or from a mass-dependent fractionation or mass-dependent system. For those of you who are not isotope nerds like myself, we express changes in the ratios of certain isotopes of sulfur with delta notation, right? So delta 34S, which is the most common sulfur isotope number you will see, is represented by this ratio of 34 to 32S in a sample relative to that ratio in a standard, right? And so this is your very typical sulfur isotope number that you'll see. Now, sulfur has more than 2 stable isotopes, so it can have mass 32, 34, but it can also exist as mass 33. And we can cast this same ratio in terms of delta 33S. Now, in a mass-dependent world, that delta 33S should be about half of our delta 34S in the same sample, right? So 33 is about half as different to 32 as 34 is, right? So 1 mass unit away versus 2, so it makes sense that it'd be about half as big. And if we look at sulfur isotope measurements after the Great Oxidation Event, that's what we see, right? So if we plot 34S versus 33S, we see this really tight linear relationship that has a slope of about 0.5, right? So it really does work out. Now, if we go prior to the GOE, these points in orange here, we see this really distinct deviation from that nice linear relationship that we saw on the previous slide. And our CAP33S values are basically quantifying that deviation. So how far off the line are our delta 33S values based on a measurement of delta 34S values, right? So when we talk about CAP33S, we're talking about this deviation from expected mass-dependent behavior. So What is it exactly that is causing this deviation? Well, you might have guessed it's oxygen, right? So if we, our best understanding of how this process works is that we have the reaction of sulfur dioxide gas with UV light in the atmosphere. So if we go back into the Archean, where we don't have any oxygen around, we can have this reaction occur, and we can split this SO2 into a reduced phase and an oxidized phase, right? And so this reduced phase is going to have positive Cap33S values, and the oxidized phase negative. And again, without any oxygen around, we can then take that reduced phase and preserve it in the rock record separate from our oxidized phase, which gets preserved as something else. And so because of that, we can then actually generate and preserve appreciable Cap33S signatures in the rock record. Fast forward to an oxygenated atmosphere. And first of all, oxygen in our atmosphere also begets ozone, and that ozone is a shield of the UV light that can cause this reaction to occur. We still do have SO2 that gets up to the stratosphere, undergo this reaction and split into this reduced and oxidized pathway. But once that S8 falls down into the oxygenated, or falls down to our oxygenated atmosphere, it transitions back to this oxidized form, they recombine, and we lose any MIF signal. Right. So the presence of appreciable Cap33S values is a really good indicator for the presence or absence of oxygen. And in general, we think the levels of oxygen needed to inhibit this process or this preservation is about 10 to the minus 5 of atmospheric levels. So pretty low concentrations of oxygen will shut down this pathway. In the ensuing 25 years after Farquhar et al. Published their paper, the measurement of Cap33S values exploded, right? So we have this enormous data set. And I know this is the Earth Data webinar, so if you want to get your hands on some of this data, you can associate with our Nat Comms paper. But we can see this, 20 plus per mil range in Cap33S values prior to about this 2.3 to 2.5 billion year mark. However, more data, as is usually the case, equals more interpretations. It's kind of muddied the waters on that strict timing of the transition to an oxidized atmosphere. And those interpretations, people going back and forth on those interpretations and making measurements in rocks all around the world lead to people kind of pulling their hair out and making figures like this, right? Trying to correlate these transitions from SMIF or mass independent world to a mass dependent world across some boundary, and trying to line all these up with various ages and be able to say, okay, is this one rapid singular event where we cross the threshold for from at mass independent to mass dependent world, or is this a back and forth? Do we see this rise and fall of oxygen across this time period? And much of the debate, it's a really complicated story, but much of the debate kind of centers around this setting in the Transvaal supergroup in South Africa. And really it boils down to the difference between these two formations known as the Deutschland and the Reuhookte, right? So to kind of simplify that diagram down, both the Deutschland and Reuhookte formations have been shown to have MIF sulfur transition into mass dependent sulfur across their stratigraphy. However, the Deutschland has a very poorly constrained age, and people have used, say, tetrhyl zircon ages and other things that are less precise to say that the Deutschland formation is equivalent to the Reuhookte, and other people have said it's distinctly older than the Reuhookte. And what this means is that if they're time equivalent, it suggests we have a singular event or one rapid rise of oxygen that doesn't go back. If the Deutschland is older, it suggests that we have this back and forth, these rises and falls of oxygen across the threshold needed to inhibit sulfur myth. Okay. And so this has been a long-standing debate in the literature. And there was about 2016, there was a massive effort, starts with the Chimera, Agaron, GOE, and Biomarker Drilling Project, where they went to 3 locations and drilled 4 cores across the Transvaal Basin in South Africa. And you can see here, the first being ANW1 in the eastern part of the basin, ADL1 in the northern part of the basin, and AGP1 and 2 in the western part of the basin. And ANW1 transects the Reuhuchte Formation, ADL, the Deutschland, and AGP1 and 2 transect the western equivalent of the Deutschland in the basin. And so we have this really kind of unprecedented spatial coverage across these formations to really go at this question of what was the nature of the Great Oxidation. It was a series of episodes or one singular event. And these can all be correlated to previous work that's been done down here in the southern part of the basin in these 3 cores. So the initial idea for how these cores related to each other is something like this. And this is going to change, so don't get too attached to this. But the initial idea is that A and W over here in the Reuhuchte was directly correlatable to the upper part of AGP2, and that ADL and AGP1 were kind of lower, but still intersecting the same equivalent formation, right? So this was the idea, and that the ADL1 was in a shallower proximal marine setting, whereas ANW1 was the more distal setting, and AGP and AG1 and 2 were somewhere in between that in terms of water depth. These cores are kind of incredible, and they were really a pleasure to work with. They intersect and transcend really different redox conditions in the basin. So even just in this one core box, you have this really stark transition from this kind of more organic enriched black shales all the way up to these really organic, lean, fine sandstones. And you can see this real distinct transition in redox and sedimentological environment. And the really other amazing thing about the placement of these cores is that they preserve really disparate sedimentary environments. So we have anything from, these coarse cobble conglomerates, so these rounded edges, a really high energy system, to these chemical sediment, this iron formation, so this really low sedimentation rate, chemical sedimentation, to these kind of more punctuated mass wasting deposits where you have rip-up clasts and these kind of high energy flows in the basin. And we have anywhere from what seem to be relatively oxidizing conditions to conditions where you can preserve these massive conglomerations of pyrite. Right, so this is all pyrite up here in this gray laminated gray shale. And so these cores really can give us a nice picture of how the rise of oxygen and other geochemical parameters change all across the basin in very different environments and give us a better idea of the kind of a holistic change in the redox state at this time. So in order to kind of get at this question of where is the Reuhuchte and where does the Deutschland fall relative to the Reuhuchte, the obvious kind of thing to go after is a new age. And that is what we did. And, I say that kind of glibly because this was a project led by Alexei Millikan, our colleague at Yale. And this was the result of, many, many hours of really painstaking geochemistry to produce this age, this isochron. And what you can see here is that in core ADL1, within the Deutschland formation, we have an age of about 2.443 billion years, or 2,443 billion years. And what that indicates is that the Deutschland is indeed older than the Reuhuchte, right? So this is that age that I just talked about in the Deutschland formation. And what you can see here is it's distinctly older by about 100 million years than any age that we have for the Reuhuchte, right? And these are direct, this is another rhenium osmium age. This is a uranium lead date on a tuff, right? So this is demonstrably and distinctly older, okay? And what that then means in the context of the GOE is that we really do have at least one massive fluctuation in atmospheric oxygen well before the permanent oxygenation of the atmosphere at about 2.31 billion years. Sorry, I have a little friend here getting to join. Right. And I say at least one because we have more sulfur isotope data associated with these cores that is on its way. So stay tuned about that. That's being led by my colleague, Gareth Izon. But in any case, this is really good evidence that we have at least one episode within the Great Oxidation event. So that tells us something about the atmosphere, right? We have this idea that this was a messy transition from an anoxic world to an oxic world in terms of atmospheric oxygenation. We also have really good evidence from the marine system. So there are a plethora of trace metal isotope proxies that indicate fluctuations in marine oxygen contents between, say, 3.2 and 2.3 billion years ago, right? So you have the kind of classic original WIF paper, so looking at molybdenum concentrations and various other trace metal systems in the Mount McRae Shale. We have, transient oxygenation of surface oceans and this whole slew of ideas of what oxygen is doing in the ocean based on various metal isotope and metal concentration proxies. So these are all the different kind of interpretations of what's going on. And figuring out which is right is, difficult and maybe not necessarily as necessary. But what is really clear here is that we have a pretty good assemblage of evidence for fluctuating redox conditions in the ocean well prior to these fluctuating oxygen conditions in the atmosphere. And so the way we're going to try to add to that or kind of dig into that story and say something a little bit more is we're going to look at nitrogen, right? And so why are we going to look at nitrogen other than the fact that that is, what I do? I'm a stable isotope nitrogen geochemist. Nitrogen is redox sensitive. So if we look at the biological cycling of nitrogen in the modern system, we have this really strong gradient in oxidation states, anywhere from -3 for its most reduced form of ammonium and organic nitrogen, all the way up to +5 for nitrate, right? So in 9 different oxidation states, all over which you can have different types of biological cycle, right? So there's a lot of potential here to trace these transformations as we are transitioning from a more reduced world into a more oxidized world. The other bit about this, aside from using nitrogen to say something about oxygen, is it's nitrogen in and of itself is interesting because it's one of these kind of fundamental, big 6 building blocks of life, right? So nitrogen is used in various lipids, amino acids, proteins, nucleic acids, RNA, DNA. It is a major component of life and the building blocks of life, and one of these things that is used by all life as we know it. So understanding how nitrogen is cycled through time and how that cycling is changing is also really interesting from a geobiological perspective. So we know that nitrogen is an implicit constraint on life and that its processes are redox sensitive, and that these processes can be traced with the stable isotope ratios of nitrogen, or delta 15N. So very similar to that delta 34S definition that I showed you before, just this case with nitrogen. And we can see this really immense scale of variability in nitrogen isotopes on the y-axis here through time. And if we want to dig into this figure a little bit, what you can see here is that if we get rid of even if we get rid of our highly metamorphosed samples that are above green schist, we see this really amazing transition from a Paleoarchean, Mesoarchean world where our delta 15N values kind of fluctuate around 0, which is roughly the input value from molybdenum-based diazotrophy, to something about a range from -11 up to 43 per mil or so. This collapse back down and this kind of variability back and forth all the way through geologic time. And so to give you an idea of the processes that are causing these variations in nitrogen isotope ratios, what I need to do is kind of put everything within a context of the framework of the modern ocean, right? So if we look at the modern range, that's this gray box here, and that can give us an idea of what's causing some of these variations and how we have to think differently about how to explain some of these other ones. So the modern nitrogen cycle looks something like this. It's a mess. This is the spaghetti or ramen or noodle diagram where everything's just kind of running together. And we can kind of take this system and simplify it down to the major processes that control the concentration and isotopic composition of dissolved inorganic nitrogen. And in this case, in the modern system, the predominant form of that is nitrate. So if we simplify this down to those two main levers, we have the input. The main input in the ocean is the fixation of N2 into diazotropic biomass, which is then remineralized into ammonium. And then ammonium is rapidly oxidized and nitrified to nitrate in the modern ocean, right? Ammonium does not stick around very long. That nitrate is then either taken up directly by organisms, or it can be upwelled through, say, an oxygen minimum zone, where it's denitrified and lost from the system as N2 or N2O. And these two processes have really distinct impacts on the concentration and isotopic composition of our dissolved nitrate pool, right? And so that is kind of our reference point for the modern system. So this is the average composition of marine nitrate in terms of concentration on the X here and delta 59 on the Y. And as you can see, our fixation process adds nitrate to the system to the right and decreases its delta 59 value, whereas denitrification removes it from the system and increases our delta 59 value. And then these other processes have varying impacts as well, but they're not as important or as widely expressed as these two main levers. Okay, and so the average rate or balance of those processes fix the composition of our nitrate, and that pins the average of our sedimentary delta 59 values. And so we can see here in the modern system, that average, which is this kind of light blue color, is pretty well distributed, but you can have localized processes that are overprinting it, right? So you can have denitrification in oxygen minimum zones picked up by these purple shading here. And you can get really positive delta 59 values associated with that incomplete denitrification. So all of this averages out to a core top delta 59 value of about 5 to 6 per mil. Prior to the rise of any oxygen, or so before there was any oxygen in the system, the nitrogen cycle looked very different. So you still have fixation by diazotrophs, but that remineralization to ammonium did not proceed to an oxidation step, right? So we didn't have this direct rapid nitrification to nitrate. We had the remineralization to ammonium, and then the fate of that ammonium was either assimilation by microbes directly, or this process known as iron anammox or Fiamox, which has a much smaller isotope effect, right? And is much less efficient. But that was the kind of main lever for removing that ammonium from the system. And so what we have here is an ammonium ocean, or a nitrogen cycle based on ammonium with some with some oxidation by iron oxides or direct assimilation by organisms. And so what we'd expect here is that our delta 15N values in this sort of system hover much closer to 0 and maybe skew up to, say, 5 or 6 per mil at most. And that's really what we see. So if we look at the Paleo to Mesoarchean relative to our modern core tops here, these are just histograms of all the measurements that I could dig out of the literature. We see this distinct, much more negative or near 0 delta 15N distribution in the Paleo Mesoarchean relative to the modern system, which is dominated by nitrate and denitrification. So now what we're going to try to pin down is how do we go from here to here? How do we go from the Paleoarchean to the Mesoarchean to the Neoarchean, the Siderian, and on down the line to our modern range, which is represented by this green box in the back. That's our modern range and modern average and the white line up and down. And what you can see is that across these time periods leading up to and after the GOE, we have this distinct rightward movement, right, in terms of the average or the mean distribution. Okay, so in our older systems, we're all generally towards 0, and we're slowly moving right and kind of going back and forth around that average. What you can also see, particularly in the Neoarchean, is that we tend to have these really extreme end member values, these -10s and -30s, and even upwards of -40 in some of these older systems. And really what we're seeing here is this transition. I'm going to simplify everything down again to the most kind of relevant points. We have this shift in a system from pre-great oxidation event, where ammonium is our main DIN form, and we have fixation and Fe anammox as the main in and out, along with uptake and sedimentation by biology. Then we move into a syn or during GOE time period where, again, fixation to ammonium, and then that ammonium is either directly taken up by microbes and converted to biomass and sedimentation, or we start to have the onset of what's known as this direct oxidative loss. So the Fe-amox process becomes less important, and all of a sudden now we are directly oxidizing this ammonium with a little bit of free oxygen, and we may have a little bit of nitrate around, right? And these processes are associated with much stronger isotope effects than Fe-amox, right? So this direct oxidative loss can have upwards of a 55 per mil isotope effect, and this direct ammonium uptake can have a much more negative effect. And so within this system, we'd expect really big positives and really big negatives in this kind of ammonium-based system. As we move to post-GOE, again, we're moving into this system where now nitrate is the dominant form and denitrification is the dominant form of loss, which has a slightly smaller isotope effect than our direct oxidative loss. And now we've also lost the negatives associated with direct ammonium uptake. So in this type of a system, we'd expect kind of a scale contraction. We lose the negative end members and move more towards what a modern system looked like, somewhere between 0 and 16. And if we zoom in on the time period between 2.8 and 2.2 billion years ago, that's almost exactly what we see, right? So around the Neoarchean, Alice Pellerin et al. Had this really interesting new paper come out where they basically demonstrated this direct ammonium oxidation and these really strongly positive delta 15N values in the Neoarchean, right? So we have this onset of direct ammonium oxidation and loss. But we also see at this time period these really, really negative values associated with direct ammonium uptake, right? So we have this shift to an ammonium ocean with some oxidation and direct uptake. Then we see our scale contraction down to somewhere around 0, and then this kind of slow rise after that. And previous papers have interpreted this rise as the actual onset of an aerobic nitrogen cycle. So we may have transitioned to a nitrate-based system by, say, 2.5, but it's if you were to expand these data out, they're very wobbly, so they vacillate back and forth quite a bit. You also have to take into account that these are really old rocks that have undergone, a whole lot of aging and metamorphism and different processes. And usually things within the green schist to sub-green schist range can see a delta 15N shift of, say, 1 to 3 per mil from their original value, right? So in reality, some of these numbers could be much closer to 0, and they're not really definitive in that way. So we need some more data. And what you can also see here is this really conspicuous gap in the record, right? This gap in our understanding of how the evolution of the nitrogen cycle progressed. And if you remember back to the first part of the talk, we can actually start to fill this gap with our new cores from South Africa. So this 2.4 billion year old Deutschland formation. All right, so if we go back to these cores that we've now taken an age from in order to understand the atmosphere and look at their nitrogen isotope values, we can start to fill in that gap. The problem with that was that these rocks had very, very little nitrogen in them. Right? So the bulk of the rocks that we saw, particularly in ADL, are these really kind of monotonous gray, low TOC, low TN shales that are really, really difficult to measure. And so we had to go back to Syracuse to work with my former PhD advisor, Chris Junium, and use a system we had developed there that is called a nano EARMS system. And this is a system specifically designed to measure, nanomoles, minute amounts of nitrogen in these kind of systems. Right, so we took our rocks and we were able to go here and make measurements. What we also did was we isolated some kerogen from these samples. This is a purely organic, well, mostly pure organic phase where we've removed all the silicate and carbonate minerals associated with the rock to get at an organic phase that has slightly higher concentrations of nitrogen and can use as a check of our bulk numbers. Anybody who's done a kerogen isolation can share my pain in the number of hours and amount of effort it took to get this tiny little vial full of kerogen. And this is actually one of the samples that had quite a bit in it relative to others. And so these are pretty precious commodities, but we were able to make it work. So our results from this study, if we look at ADL and AGP first, and again, remember our Deutschland formation sits down here below the Reuhuchte, we see this really incredible delta 15N record, particularly in ADL1. Right, so just to give you a sense of scale here, we're going from 0 per mil in nitrogen space to about 20 per mil across the length of the core. And if you remember back to our modern range, it ranges from about 0 to 16. So we're beyond the modern range in just one core, and in this kind of continuous upward heavying. What we see in AGP 1 and 2, which is slightly deeper in the basin, everything sits at around this kind of 5 to 10 per mil range. And our kerogen numbers in the triangles here track them as we'd expect them to. And so we have this really distinct heavy values in our Deutschland formation. What we also see in ANW1, which we can correlate to previous studies that have looked at delta 15N values across this time period, if we correlate to a common surface, we have pretty comparable values. And what this kind of tells us is that all across the basin at this time, our kind of our dissolved inorganic nitrogen pool was pinned somewhere between, say, 4 and 10 per mil, and it was pretty consistent. What we can also tell from our ADL and AGP records is we see this ingrowth of progressive denitrification in the basin, right? So we have this deep to shallow gradient in nitrogen processing, which we take to indicate this progressive denitrification process that today we see across oxygen zones. So the spatial aspect is also really interesting in that regard. So what we can kind of say is that we have really strong evidence for what we think is an oxidized or an aerobic nitrogen cycle associated with this first initial episode of atmospheric oxygenation in this basin. We wanted to take that a step further and do a little bit of statistics. So here's that same record in context through time. And we wanted to see, with a little bit of statistical work, can we pin down that exact timing of the aerobic nitrogen cycle? And I know I'm getting low on time, so I'm going to kind of jump through this. But basically, in this diagram, the valleys here, these negative peaks, are associated with Nick points or pivot ages in the nitrogen cycle based on delta 15N values. And so we can see the onset of ammonium oxidation and this transient oxidative end cycling. We see another peak, or I should say negative peak, in the nitrogen cycle concurrent with our new delta 15N data from the Transval. And this pushes back the previous peak that used to be around here at 2.3 as reported by Zirkle. So we can really pinpoint this onset of the aerobic nitrogen cycle with this new data set. What we think this means in terms of the oxidation of the Earth's surface is this kind of vacillation back and forth between aerobic and anaerobic cycling may have something to do with this push and pull between the biosphere and the geosphere as organisms kind of gradually evolve the capability to overcome oxidative stress. So this is an idea that was put forth by Woody Fisher et al. About 10 years ago now, because when oxygen was first beginning to accumulate, it would have been toxic, right? And so organisms had to come up with a way to deal with that oxidative stress. And so trying to make use of oxygenic photosynthesis, which is the most energy efficient form, while also being able to deal with the toxic byproduct. And so this back and forth may have had some impact on this kind of vacillating oxygen contents. The other kind of part of this, similarly to nitrogen, is that nitrate is not ammonium, obviously, right? It's a different redox state. But in terms of biological requirements, life evolved under an ammonium ocean with the capabilities to intake and utilize ammonium to make various biochemical components. And most organic nitrogen is in the same oxidation state as ammonium, right? So taking that directly in and putting it right into your glutamate and other cycles. Once we transition to a nitrate ocean, now organisms have to evolve the ability to not only take in this oxidized anion, they have to reduce it across 9 oxidation states all the way to ammonium in order to utilize the rest of their biochemical machinery. And so we can see this kind of as an evolutionary threshold that organisms had to figure out as we move towards a more oxidized end cycle. So conclusions, the permanent atmospheric oxygenation around 2.3 billion years ago was precluded by at least one episode. There may be more episodes. The nitrogen cycle had transitioned to an aerobic state by that time, and the dissolved inorganic nitrogen pool was dominated by nitrate rather than ammonia. These are kind of the big takeaways from this study. I want to take a second to thank my co-authors, Gareth Eison, Shuhei Ono, and Roger Summons at MIT, Alexei Millikan at Yale, who did the RIAS work, Chris Junium at Syracuse, who provided the access to the nano, and the late Nick Bucas, who was the one who really led the charge for getting these cores drilled and out of South Africa. And he will be greatly missed in the geochemical community. Thank my funding sources, and I really want to thank you all for having me. And I'd love to take any questions. If you want to see more, here's my website.
Now is the time for questions from our participants. So feel free to open your mic to ask a question, or you could leave the question in the chat and I could read the question on your behalf. Okay, any questions? Okay, let me start with one question. Out of curiosity, that from my perspective, that for the Archean atmosphere, if it is in reduced form, then it's likely that ammonia uh, uh, is stable in the gas. So does that make difference from transited from the nitrogen gas to the to the reaction path that you wrote on earlier, or it is started from ammonia in the gas and continue with the reaction that you mentioned?
So it could. So absolutely. So you could have some ammonia interactions with the marine system. What's kind of hard to pin down in that reaction is the impact of pH, right? So from the marine perspective, the transition from or the volatilization of ammonium to ammonia has a pKa around, I think it's like 9.2 or 9.5, somewhere in there. And so you have to have relatively alkaline conditions in order to drive the volatilization of ammonium to ammonia. And there are systems, so like the Tumbiana Formation has been kind of recategorized as this shallow alkaline lake that really you can see evidence of this process going on, not only in nitrogen isotopes, but in various geochemical proxies of that lake. So you absolutely can have this transition from ammonium to ammonia. Now the reverse, I'm less well versed in, and I don't really know how much ammonia was being produced or what the concentration of that ammonia was in the atmosphere. My understanding is it's not that high, but maybe you know better than I would. But certainly from a marine to atmosphere perspective, you have to have the right setting for that to occur.
Mm-hmm. Okay, that's interesting because from my perspective, that the Earth's initial atmosphere started from the magma degassing. So it could start earlier on from the magma ocean degassing to the volcanic gas degassing. So to me, that the initial nitrogen isotopic concentration in the atmosphere started from the Earth's mantle, for example. But according to your interpretation, I think it's quite interesting because this is must be if no matter what's the starting gas composition, what we see in the sediments basically is a combination of oxygen oxidation in the ocean as well as the pH values.
Yes.
So
yes.
So for those plots, let's say a combination of both pH and oxygenation plots that could narrow down the possible pathway that we are seeing.
Absolutely. And in terms of trying to get at this, really early, magma ocean, this degassing and this ammonia, one of the things we're kind of limited by is our ability to make measurements of nitrogen isotopes past, say, like 3.5, right? So I don't necessarily know the full like transition, but trying to get actual measurements of rocks from that age is really difficult. Outside of the odd zircons and like the Jack Hills or something like that, but trying to get nitrogen isotope measurements back then would be difficult. So I think that would be it'd be a modeling problem, which I'm sure you're much more suited to handle.
Okay, thank you. Next question, please.
Hi, hi, can you hear me?
Yes, hi.
Hi, Ben. I'm Yongdu from China University of Geosciences. Thanks for sharing. I have a question. Can the Phanerozoic have the similar situation as the pre-Geology? We know that the Phanerozoic have a very high oxygen level. Uh, you, but there's some time during the the OE event OE during the OE can the ocean become similar like pre
Uh,
GOE? The
yes, absolutely.
I mean the deep ocean, the deep ocean totally is also dominated by ammonium.
Yes, absolutely, and that's something that I've also worked on a fair bit with my PhD advisor Chris Junium and my colleague Ann Pearson at Harvard, and looking at. I'm on the perfect slide for this. We can zoom in, and these negative spikes that you see here are almost all from some form of interpreted oceanic anoxic event, right? So here's the classic OE2 negative spike, and generally what the thinking is behind um, some of these really negative values um, is that we are building up some deep ocean reservoir of ammonium in these really reduced basins during oceanic anoxic events. So you have this really strong redox stratification where your deep ocean is anoxic and you can build up some degree of ammonium, and then you can go through, say, these periodic deliveries of that ammonium to the surface ocean where organisms can make use of that ammonium and drive these really negative isotope values or delta 15N values associated with those OEEs. And that's something we've coined as the ammonium nutrient capacitor for oceanic anoxic events. We've got a paper in geology about it. I can send that to you if you shoot me your email. And we very think about yeah, this kind of reemergence of ammonium as a nutrient source in these specific isolated basins during the Phanerozoic, for sure.
Yes, I have seen the paper. The paper, yes.
Okay.
But I think this is maybe regional, not the overall ocean.
Oh, yes, I would agree with that.
Yes, the overall
Um,
ocean become ammonium dominated is really un, unbelievable. I
no, yeah,
think.
I totally agree. And using like OEA2 as the example, like the type example, really. So if you look at all of these numbers here, and it's mostly driven by the proto North Atlantic, right? So you have this kind of relatively restricted, enclosed, large ocean basin, but it's not the full ocean at that time. It's a restricted subset of the ocean where you have the conditions that are conducive uh, to driving this redox stratification and building up that ammonium. So I totally agree with you that it's going to be in isolated basins, and probably much of this average curve that you see here um, is where it's dipping down like this is driven by the fact that just so many people have made measurements of delta 15N in OAEs. Everyone's chasing that oceanic anoxic event nature paper, right? So that's kind of biased in this particular record.
Yes, yes. Thank you.
Sure. Thanks for your question.
Okay, next question. Okay, I think that's okay. Thank you very much, Ben, for keeping you so late. There's 12-hour difference. It's already 10 p.m. At Cornell. And thanks everyone for your participation. So this video will be shared on YouTube for outreach for science. Actually, there are many more times that people watch the videos than people are going to watch the live conversations that we're having right now. So thank you very much, Ben. And will you go to Goldschmidt this year?
I'm not sure. I have to look up and see if there's a session that I can still make a play for, but I hadn't planned for it, but we'll see. I might change my mind.
Okay, okay. I will go. So maybe I will see you in person. Okay,
All right,
thank you
I'll
very
look for
much.
you if I go.
Yeah,
All
thank
right.
you, everyone. Bye-bye.
Thank you very much for having me. Bye.